Hi, I'm Jenny. Welcome to my channel, My Guruji. Today's class is based on neurobiology, where I'll be taking the outline of the nervous system and the cells included in the nervous system and its functions. So, the nervous system, as you might know, is divided into the central nervous system and the peripheral nervous system. The central nervous system consists of the brain and the spinal cord. The peripheral nervous system consists of all the nerves that feed stimulus into the central nervous system. The cells of the nervous system includes the neurons, which are the basic functional units, and the glial cells, which are the supporting cells. So as you can see in this figure, there are two sections of your nervous system, the central nervous system and the peripheral nervous system. And the central nervous system, you can see that input comes from the peripheral nervous system to the brain and spinal cord, and output goes to the peripheral nervous system from the central nervous system. So the input stimulus is received from sensory and visceral means, that is known as the afferent division, and the output signals are the efferent division. So the output division or the efferent division includes the somatic nervous system and the autonomic nervous system. The somatic nervous system consists of motor neurons, which innervate skeletal muscles. So in fact, it is important for your voluntary movement of your muscles. The autonomic nervous system can be divided into three different sections, that is the sympathetic nervous system, the parasympathetic nervous system, and the enteric nervous system. The enteric nervous system receives stimulus from the digestive tract and innervates the digestive organs. The other two, the sympathetic and the parasympathetic nervous system, innervates the smooth muscle, cardiac muscle, exocrine glands, and some endocrine glands. So this is the overall outline of the central nervous system and the peripheral nervous system. Moving on, we'll go into the neurons. So the neurons, as you can see in the figure, they're highly specialized, interconnected, and highly differentiated cells of the nervous system. They have a speciality in the fact that they are bioelectrically driven, and the size of the neurons are variable, ranging from 5 to 8 microns in diameter to 120 microns in diameter. As you can see in the figure, the neurons are divided into two major sections, the cell body and the axons. So the parts of a neuron, the neurons have an axon, which is often branched, and it transmits signals to interconnected target neurons. The dendrites extend from the nerve cell body and usually branch in extremely complex patterns. They have multiple short protrusions called the dendritic spines. The cell body is also known as the soma or the perikaryon, and it contains the cytoplasm surrounding the nucleus. And the function of the cell body is to receive synaptic contacts from other neurons. So neurons can be classified in several ways. One of the classification is based on the function, where it is classified as sensory or afferent neuron, interneuron, and motor and or efferent neuron. The afferent neuron receives stimulus and passes it to the central nervous system. The efferent neuron receives stimulus from the central nervous system and passes it to the effector organ. The interneurons act as connections between the afferent and the efferent neurons. So the afferent neurons are usually sensory neurons which have a sensory receptor which can receive stimulus from the different sense organs of the body. Whereas the motor neuron innervates the skeletal muscles or glands for voluntary movement. Another classification is based on the structure, where it can be classified based on the position of the cell body. If the cell body has a single neurite, a neurite is actually an extension from the cell body of the neuron. And if there is a single neurite or a single extension from the cell body, that neuron is known as a unipolar neuron. If there are two extensions, one being the dendrites, the other being the axons. If there are two extensions, it's called a bipolar neuron. If there are multiple extensions, where there is a single axon but there are several dendritic extensions from the same cell body, it's called a multipolar neuron. So these are the normal types of structural differences, but there are some special characteristics of specialized neurons. One example is a pseudo unipolar neuron. It's called pseudo because it's false. It looks like it's a unipolar neuron, but it actually has more than one extensions from the cell body. Another type of neuron is the anaxonic neuron, which does not have an axon. It has several dendritic extensions from the cell body, but no axon. Purkinje cell is another type of multipolar neuron, where the extensions from the cell body, which forms the dendrites, forms a tree-like reticular structure, and the axon terminal forms a triangle-shaped structure. The last one is a pyramidal cell, where the cell body is shaped as a pyramid. Again, it's a multipolar neuron because it has several projections or extensions from the cell body. So next is the glial cells of the nervous system. The glial cells are also known as neuroglia, and they support the interneurons physically, metabolically, and functionally. 90% of the cells are found within the central nervous system. The glial cells do not initiate or conduct nerve impulses, and they help support the neurons both physically and metabolically. The glial cells are also important to maintain homeostasis of the environment surrounding the neurons, and they can actively modulate, that is, they can depress or enhance synaptic function. And the glial cells are as important as neurons for learning and memory. So there are four major types of glial cells in the central nervous system: the astrocytes, the oligodendrocytes, the ependymal cells, and the microglia. So we'll start with the astrocytes. The astrocytes, as the name suggests, they are star-shaped and most abundant of the glial cells. They have several critical functions, including physical support for the neurons. They serve as scaffolds during fetal brain development. It can induce formation of blood-brain barrier, helps in the transfer of nutrients to the neurons, forms neural scar tissue, takes up and degrades released neurotransmitters, takes up excess potassium to help maintain homeostasis of extracellular fluid ion concentration, enhances synapse formation, and strengthens synaptic transmission. So astrocytes are also important for communicating with neurons and among themselves. Second is the oligodendrocytes, and in this figure you can find the differences between an oligodendrocyte and an astrocyte. The astrocyte has several projections at the outset. The oligodendrocytes also look the same, but the astrocytes have a connection to all the different cells. They have a connection to the neurons, they have a connection to the ependymal cells, and they also have a connection to the capillaries. So it actually acts as an interface between the capillaries and the cells of the cells of the nerve system. And the oligodendrocytes also have several projections, but the projections, as you can see, is found encircling the axonal areas, whereas the astrocytes do not encircle the place that they bind to. So the oligodendrocytes forms the myelin sheath in the central nervous system. That is, the projections wrap in a jelly roll fashion around the section of a axon to form a patch of myelin. So later in fetal life, oligodendrocytes start to begin secreting nerve growth inhibiting proteins, also called NOGO, and the nerve growth inhibitors actually serve as guardrails which helps keep the nerve endings from straying away from their normal positions, and this helps to stabilize the complex structure of the central nervous system. One of the disadvantages of inhibition of growth by the growth inhibiting proteins is when the central axons are damaged and require a repair. The next cell is the ependymal cell. So as you can see in the figure, the ependymal cells line the nerval tissue or the nervous tissue, and it looks somewhat similar to the epithelial tissues. So it basically lines internal fluid filled cavities of the central nervous system, and the cells lining ventricles help form the cerebrospinal fluid, serve as neural stem cells which can form other glial cells and new neurons. So new neurons can be produced with the help of ependymal cells which act as neural stem cells, but the fact that should be noticed is that the new neurons are only produced in a particular part of the hippocampus in the brain. Neurons in the rest of the brain is considered irreplaceable. So why are these ependymal cells normally dormant? How can they be activated? And what if the activation of these ependymal cells leads to the possibility of self-repair by the brain? Think about it. And finally, there's the microglia. So the microglia are actually immune defense cells of the central nervous system, and they remain stationary until it becomes activated by an infection or an injury. And activated microglia are highly mobile. That is, it can move towards the affected area to remove any foreign invaders or tissue debris by phagocytosis. And they can also release destructive chemicals against these targets. Again, overzealous microglia or excessive action of microglia can cause damage to the neurons, and they contribute to neuronal damage, as that can be seen in stroke, Alzheimer's disease, multiple sclerosis, dementia of AIDS, and other neurodegenerative diseases. So these were the 4 cells or glial cells of the central nervous system. Next is the glial cells of the peripheral nervous system. So Schwann cells are one of the cells in the peripheral nervous system, and they are important for the myelination of the neurons in the peripheral nervous system. There are in fact two types of Schwann cells: myelinated Schwann cells and unmyelinated Schwann cells. And in this figure itself, you can see that in the myelinated Schwann cell, the Schwann cell wraps itself around the axon in between the nodes of Ranvier to form several concentric layers, and the cytoplasm of the Schwann cell, including the nucleus, gets pushed towards the outer edge of the Schwann cell. Whereas in unmyelinated Schwann cells, even though it surrounds the axons, it does not wrap itself around to form a sheath. So the Schwann cells are also known as neurolemmocytes, and they are called the myelin forming cells of the peripheral nervous system. They can also form regeneration tubes and secrete nerve growth enhancing proteins, which guide and promote regrowth of damaged peripheral axons. The cell body and the dendrites should remain intact for regrowth of damaged axons. Permits return of sensation and movement. And the myelinating Schwann cells are usually found around the axons of motor and sensory neurons. So this is the structure of myelination, or the figure for myelination. Finally, we have the satellite cells, which are flat neuroglial cells which surround the cell bodies of the peripheral nervous system, provides structural support, regulates the exchange of material between the neuronal cell body and interstitial fluid, usually nutritional material, act as protective cushioning cells, and express a variety of receptors which allow for interactions. So this is the structure of a satellite cell, which is surrounding the cell body. And we will go into a small medical information, that is, the glial cells do not lose ability to undergo cell division. So most brain tumors of neural origin are caused due to the glial cells and are called gliomas. Neurons themselves cannot form tumors because they do not have the capacity to divide and multiply. So brain tumors of non-neural origin are of two types: those that metastasize to the brain from other sites, and those that originate in the meninges, called as meningiomas. Functions of central nervous system: regulates your internal environment, helps you to experience emotions, controls your movements, makes you aware of your own body and your surroundings, and engage in higher cognitive processes like thought and memory. So the central nervous system is protected from injury by four major features present in your body. One is the hard bony structures, cranium or the skull, which encases brain, and the vertebral column, which surrounds the spinal cord. Second is the meninges. Three protective and nourishing membranes are present between the bony covering and the nervous tissue. The cerebrospinal fluid, which is a special cushioning fluid, and the highly selective blood-brain barrier, which regulates exchange between the blood and the brain. So the blood-brain barrier constitutes of cells that form walls of a brain capillary, which are joined by tight junctions, and these impermeable tight junctions seal the capillary wall. Transport across the brain capillary walls between the wall-forming cells is anatomically prevented and transport through the cells is physiologically restricted. Functions of the blood-brain barrier: protects brain from chemical fluctuations, minimizes the possibility of harmful substances reaching the central neural tissue, prevents certain circulating hormones or neurotransmitters from reaching the brain. The disadvantage of the blood-brain barrier is the fact that it limits the use of drugs for the treatment of brain and spinal cord disorders. The cerebrospinal fluid is a clear colorless body fluid found in brain and spinal cord, and it's produced by specialized ependymal cells in the ventricles of the brain. The cerebrospinal fluid circulates throughout the ventricles and the central canal over the entire surface of the brain and the spinal cord. And the cerebrospinal fluid has the same density as the brain itself, and the brain is essentially suspended in this special fluid environment. The major function of the cerebrospinal fluid is that it acts as a shock absorbing fluid to prevent the brain from bumping against the interior of the skull in case of sudden jarring movements. Finally, the functions of the peripheral nervous system: it actually acts as a connection that connects the central nervous system to the organs, limbs and skin. It allows the brain and spinal cord to receive and send information to other areas of the body, carries sensory and motor information to and from the central nervous system, regulates involuntary body functions like heartbeat and breathing. So, we have come to the end of today's class. Thank you for joining me. If you have any questions or suggestions, you can comment below in the comment box. If you like this video and would like to hear more such video lectures, please click the subscribe button and the bell icon below. Remember, one step every day.